Hi guys, this is Biochemistry 2 Presentation Group, Sub S. This is first presenter Yejin. First, why is this called the citric acid cycle or TCA or Krebs cycle? This chemical pathway is called the citric acid cycle because first intermediate compound formed in this cycle is citric acid. And why TCA? It is because first intermediate, which is citrate, is also called tricarboxylic acid. And for the last, why Krebs? It's because who explained this cycle is Hans Krebs. He is also best known as citric acid cycle and also urea cycle. And why is this chemical pathway so important in the metabolism of cells? It is because it produces most of the energy from the oxidation of glucose and fatty acid, producing ATP, which act as energy currency of the cell, and it is considered the most important component in cell. Third, why is this pathway so complicated? Why does this pathway take the form of a cycle? And is this pathway efficient and smartly designed? Yes, cyclic form is to continuously oxidize acetyl-CoA and recycle byproducts to increase efficiency. Like I marked here in the picture, in result, this form extracts energy through step-by-step reactions and maximizes energy efficiency. So fourth, I'll explain briefly the entire citric acid cycle. Pyruvate is oxidized to form acetyl-CoA, which initiate the citric acid cycle. And acetyl-CoA, which has 2 carbons, combined with oxaloacetate to form citrate, which has 6 carbons. This reaction is irreversible. This cycle begins with the formation of citrate. Next, citrate then undergoes an isomerization reaction to become isocitrate, which has the same number of carbons. Isocitrate forms alpha-ketoglutarate, which has 5 carbons, releasing CO2. During this process, NAD+ is reduced by gaining electrons. This reaction is also irreversible. Next, alpha-ketoglutarate makes succinyl-CoA, which have reduced carbons, and it releases CO2, and it's irreversible too. 3 irreversible reactions are in this cycle. From now, there is no change in number of carbons. Succinyl-CoA forms succinate, which doesn't change any carbons, and it makes ATP or GTP. Succinate forms fumarate and reducing FAD to FADH2. Fumarate makes malate and reducing another molecule of NAD+ in the process. Lastly, malate regenerates oxaloacetate, allowing the cycle to begin again with another molecule of acetyl-CoA.
Hello everyone, this is the second presenter Minji, and I am going to talk about a disease named very-very. Very-very is a disease caused by thiamine deficiency, and it directly affects the citric acid cycle. Thiamine, also known as vitamin B1, is a vital coenzyme in the form of thiamine pyrophosphate, also called TPP. TPP is required by key enzymes involved in glucose metabolism, including pyruvate dehydrogenase and alpha-ketoglutarate dehydrogenase, which are both essential in citric acid cycle. When thiamine is deficient, pyruvate cannot be efficiently converted into acetyl-CoA, and alpha-ketoglutarate cannot be converted into succinyl-CoA. As a result, both pyruvate and alpha-ketoglutarate accumulate in the blood, especially after a carbohydrate-rich meal that increases glucose metabolism. Since these steps are also irreversible and energy-producing reactions, the energy output from the cycle is reduced, which leads to symptoms of fatigue and weakness. There are 2 major types of symptoms depending on which organs are affected. In wet very-very, cardiovascular symptoms appear, such as rapid heart rate, leg swelling, and heart failure. In dry very-very, the nervous system is affected, which leads to muscle weakness, numbness, and coordination problems. Fortunately, the disease can be treated and even reversed by restoring thiamine levels through vitamin B1 supplements and adjusting the diet. Early diagnosis and treatment can prevent permanent damage. In summary, thiamine is crucial for the proper functioning of the citric acid cycle, and its deficiency blocks 2 important steps, causing metabolic buildup and energy failure.
The cycle begins as acetyl-CoA combined with oxaloacetate to form citrate. This condensation reaction incorporates two carbon atoms from acetyl-CoA into the cycle, enabling them to be oxidized for energy production. It creates a six-carbon compound, setting the stage for systematic breakdown and energy extraction. Next, citrate is rearranged into isocitrate through an intermediate called cis-aconitate. This reaction, catalyzed by the enzyme aconitase, involved dehydration followed by rehydration. His isomerization shifts hydroxyl group to a new position, making the molecule more suitable for oxidation in the next step. Although the standard free energy change is 13.13 kJ per mole, technically non-spontaneous, the reaction proceeds due to cellular condition. According to Rule 12, actual concentration in the cell dictates direction. Since isocitrate is rapidly consumed in the next step, its low concentration pulls the reaction forward. Also, a part will tend the removal and/or addition of water and cis-trans reagent can occur without requiring extra energy input. Now, isocitrate undergoes oxidative decarboxylation to form alpha-ketoglutarate. This multi-step process is catalyzed by isocitrate dehydrogenase and proceeds through the intermediate as oxalosuccinated and transient anode. First, isocitrate is oxidized to oxalosuccinate, transferring high-energy electron to NAD+ or NADP+. Oxidation reactions like this release energy by transferring electron and proton to electron carriers. To convert stored energy into NADH or NADPH. This reduced carrier later fuel ATP production via to electron transfer chain. Next, oxalosuccinate undergoes decarboxylation, releasing CO2 and forming an anode intermediate. This is facilitated by Mn2+, which stabilizes the transition state by interacting with the carbonyl group. Metal ion can act as electron sink to stabilize intermediates and promoting reaction like decarboxylation to reduce the carbon count and stabilize reaction intermediate. This prepares the molecule for further processing while extracting energy-rich electron. Finally, the anode rearranges to produce alpha-ketoglutarate. Alpha-ketoglutarate undergoes another oxidative decarboxylation, producing succinyl-CoA and CO2. This step is catalyzed by the alpha-ketoglutarate dehydrogenase complex. A carboxyl group is removed, NAD+ is reduced, the NADH and CoA-SH attach to form a high-energy thioester bond. Thioesters store large amounts of energy which can later be released through hydrolysis. To extract more electron and trap energy is a usable form. NADH contributes to ATP production while succinyl-CoA stored energy for the next step, substrate-level phosphorylation. The enzyme complex also uses charge functional group to stabilize the transition state. This charge group lowers activation energy, making the reaction more efficient.
In step 5, succinyl-CoA changes into succinate catalyzed by succinyl-CoA synthetase. Let's look closely at this step. This is an overview of step 5. The purpose of step 5 is to recover energy in the form of GTP through substrate-level phosphorylation. Succinyl-CoA is a high-energy intermediate containing a thioester bond, and it reacts with inorganic phosphate under the catalysis of succinyl-CoA synthetase. In this reaction,
Pi acts as a nucleophile, while the carbonic carbon of succinyl-CoA acts as an electrophile, forming a new bond. This process corresponds to chemical rule 6. When the CoAs group is replaced by Pi, a functional group exchange occurs, which is explained by chemical rule 7. Succinyl phosphate remains bound to the enzyme, and then succinate is released. The phosphate group is then transferred to the histidine residue of the enzyme. Finally, this phosphate is transferred from the enzyme to GDP, generating GTP. In step 6, succinate is oxidized to fumarate with the help of succinate dehydrogenase, and FADH2 is produced in this process. The purpose of this step is to produce energy FADH2, which stores energy in the form of high energy. Succinate undergoes dehydrogenation and is oxidized, transferring its electrons to FAD, resulting in the formation of FADH2. Therefore, this step is an oxidation-reduction reaction, and chemical rule 2 is applied. Additional FADH2 plays an important role in ATP production in the electron transport chain later. In step 7, fumarate is converted into air malate by the addition of a water molecule with the help of fumarate. The purpose of this reaction is to structurally prepare the molecule for the next oxidation step. By adding a hydroxyl group, the double bond of fumarate is broken, and a hydrogen ion is added to form air malate. This reaction corresponds to chemical rule 10, as the structure is reorganized simply by the addition of a water molecule without requiring additional external energy. Finally, in the last step, air malate is oxidized back to oxalate state, the starting point of the cycle, and NADH is produced. This reaction is catalyzed by air malate dehydrogenase. The purpose of this step is to regenerate oxalate state to restart the cycle, and to transfer 2 hydrogen ions and 2 electrons to NAD+, forming NADH. This is an oxidation-reduction reaction explained by chemical rule 2. An interesting point about this step is that although the reaction has a positive Gibbs free energy change and is thermodynamically unfavorable, the reaction proceeds in the forward direction. This is because oxaloacetate is continuously removed by the straight synthetase reaction in the next step, keeping its concentration extremely low. As a result, the equilibrium shifts towards oxaloacetate production, which corresponds to chemical rule 12. Through these processes, the stereogases cycle completes one proton and becomes ready to accept another acetyl-CoA molecule.
Those are our roles in this presentation. For quiz number 1 to 5 were assigned to Minjae and Yejin, and for question 6 is to Eunhae and Sulgi. And PPT sources and speaker are all of us, and editing the video is assigned to Minjae and Yejin. Thank you for listening to our presentation today.